Okay, so for this lecture, we're going to look at digestion and absorption of fats. So we've already talked about what fats are, some basic fats, triglycerides, phospholipids, and now we're going to go into, all right, so when we take these in, what happens from eating our fat to ultimately absorbing that in our blood. Now, if you follow along with the slides, there's a lot of information here. There's a couple pictures you can look at. I'm actually going to kind of draw some of this on our screen here. Usually in class, this is where I draw this on the whiteboard. So you can follow along with some of these slides. There are some good information here, but we're going to try to make this a little bit more real, I guess. We'll try to do this a little more digitally. So here we go. I'm going to draw right here. And again, you can butcher my not incredible artistry here. Forgive me there. So this is our mucosal cell we looked at, remember, way back when we talked about carbohydrates. All right, and what we need to do right here, this is the small intestine side, and then let's draw some blood here. So this is our blood, all right, it's our bloodstream, and ultimately we're gonna get our fats. We'll make those, there we go. We want to get our fat right here. Okay, ultimately in through our mucosal cell into the blood. Unfortunately with fats are a lot different than carbohydrates. Fats are fat, and if you know anything about fats, they are not water soluble. Okay, we often say fats are hydrophobic. And you know this if you've ever seen, it's like salad dressing, and salad dressing that sits for a while, there becomes a separation in the fluid and the other stuff in there. So we get that fats don't do well in an aqueous environment. Well, unfortunately, our small intestine is all aqueous environment, not good. And so simply just breaking fats down and then entering the small intestine really doesn't work. So in order to break fats down, it actually starts in the mouth, real to a very small extent. There's salivary lipase that's released there. In the stomach, we have a gastric lipase, which breaks down a little bit of these fat globules. But really, a lot of the work is going to occur in the small intestine. In fact, the vast majority of the work occurs in the small intestine. And some of it occurs before we even get to this point right here of this fat globule entering into our intestinal cell. A few things happen. One, the pancreas releases lipases, which are going to play a huge role in breaking these down. A second thing that happens, probably more important, is the release of bile. And so bile plays a huge role in allowing fats to be able to be broken down. We use the word emulsified, so that they can travel in this aqueous solution, be broken down by lipases, and ultimately enter into the mucosal cell. Bile works because it has a hydrophobic and a hydrophilic side, and because it had, instead of the fat just globbing together, it's able to separate it into parts. So bile has a part that likes the water, and it has a part that likes the fat. Good way to think about it. And so bile is going to work to emulsify our fats and get them to a point where they can get to the small intestine and be in small enough pieces, I guess, to enter into our small intestine, small intestinal cell. Now, another neat thing here as far as fats go is, unlike carbohydrates, remember carbohydrates had transporters on your mucosal cell? Well, fats are fat soluble. And if you remember back to biology, the cell wall here it's a phospholipid bilayer, has lipid. So these fats don't need transporters. In fact, they are just literally going to enter right into our mucosal cell. And so quite literally, they're just going to come right through and right in there. So no transporters needed. Now, these do have to be small enough, and so you have to have bile to emulsify these. You need lipases to break these down. And in fact, because of that, those two things can have an influence on how well this process works. But unfortunately, for most of us, it works really well. So these these triglycerides get broken down, emulsified by bile, broken down by lipases, made into basically smaller little globules there, and they are just going to enter right into our mucosal cell. Now, when they get into the mucosal cell, they don't simply just go right into the blood. There's actually a process where these now smaller fatty acids actually get put back together. And you actually know this. If you remember way back in Bio 1, you studied cell and cell structure. There are some structures in the cell that package things. If you remember what those are, anybody? Maybe not. One of the structures that does this is the small endoplasmic reticulum. If you remember that, the small endoplasmic reticulum. So it's going to take these small fatty acids, repackage them. From the small endoplasmic reticulum, it'll go to the Golgi apparatus. It's like high school biology all over again. Fun times, puberty, great stuff. That is going to repackage these fatty acids back into larger triglycerides so that they can then be transported out of the mucosal cell. Now, the small endoplasmic reticulum and the Golgi apparatus doesn't do this with all fatty acids. In fact, some of the smaller ones, some of these small ones, which we call small chain triglycerides, or even medium chain triglycerides. That's important. We'll talk about those this week. Those are actually going to pass right through into the blood. But these bigger ones are not. All right? They are not going to pass through. They will leave, be transported out of the mucosal cell, but they're too big to enter into the bloodstream. And this is where, when we talk about absorption of fats, something unique. There's a unique system here. And typically we make this green. I don't know why. It's not green in real life, but we'll color it green. This is where we have lymph vessels, or lymphatic system. And so we're just going to kind of draw that in green, even though it's not green. We'll do that anyway. And lymphatic system becomes sort of an intermediary means to transport these larger triglycerides throughout the body without them immediately going in the bloodstream. Now, one of the big benefits for this is that, well, think about it. We don't really want massive amounts of fat to be immediately traveling into our bloodstream. I mean, think of the damage of the blood vessels that could cause. That would be horrific. I mean, just think if you just had a really high-fat meal, you decide to eat some butter because you can, or whatever it is, that would go right into the bloodstream, clog arteries. That would be bad. And so what the lymphatic system does, it allows these to be transported, and ultimately they do empty back into the bloodstream quite a bit later, but it allows to even out these triglyceride concentrations in the blood. And this use of the lymphatic system is very different. We don't see this with carbohydrates. We don't see this with proteins and amino acids. We do see it here with fats. And it's a little different process here. Ultimately, these are going to be transported through the blood. Now, the transportation is also a little bit different. And so let's kind of go through the transportation here, because as we see with fats, triglycerides, they slowly go into the lymph here. Fatty acid concentration increases. But this is where we start to enter into a new topic that becomes really important in nutrition. And this has to do with lipoproteins. Now, lipids are generally transported in the blood via a carrier, because again, they are hydrophobic. The blood is an aqueous solution. So they need proteins to transport these. Now, you may not know much about these proteins, but you may have heard of their acronym. This is where LDL and HDL cholesterol has been really popular. And I'm almost certain every one of you has probably had a blood test at some point in your life where they told you what your LDL and HDL levels are. Well, what these are quite literally lipoproteins in the blood that are transporting our fats to wherever they're going. Now, for the purpose of nutrition, we're not going to get so much into the chemistry here. For the purpose of nutrition, what I want you to understand is that there are HDL, high density lipoproteins, which are just that, high density protein. And they actually aid to transport cholesterol to the liver. And the liver, which actually originally deals making bile with, it makes bile and then sends bile to the gallbladder to send it to the small intestine. The liver also acts to then deal with these fats and remove them from the blood. And it's a good thing. In fact, there's research that says if you have high HDL, that actually is a good thing when it comes to heart disease. You are less likely to have heart disease. So HDL, awesome. There are also LDLs, what we call low density lipoproteins. All right? These are, just like they say, low protein, but a lot of cholesterol, a lot of fats. This is bad. In fact, those who have high LDL levels, that is associated with heart disease and atherosclerosis and blocking of arteries. LDL, bad, bad. And so these proteins have lipoproteins have a huge part in nutrition, in health, in disease. There's even VLDLs, very low density lipoproteins, which is very bad, as you can imagine. And so each of these, when it comes to nutrition, is really, really, really important. And so what I want you to know is the difference between these two. And your book goes into this a little bit, but understand that what HDLs are, we often call them good cholesterol. It's not, I mean, it's true, kind of, but they're really just a lipoprotein that transports fats a little differently. We have LDL, we generally call that bad. They transport cholesterol to the walls of arteries. Each of these has huge implications as far as disease. Right? Know those. I think that's it for this aspect. Yeah, ketone bodies, we'll cover that in class. There are some pictures here I want you to look at. As I've mentioned before, I like things visual. And so for some of us, probably be able to visualize, for example, where these fats are going to go here from the lymphatic system, ultimately into the circulation. And then depending on the transporter, they're either going to skeletal muscle. Skeletal muscle can use that to form ATP. Cardiac muscle, adipocytes, that's where we store fats. Liver will transform them into something else. So again, there's some of these pictures here that help you understand this process. For your exam, yes, you have an exam coming up. Make sure when it comes to this topic, you can explain how fats are digested, how they're absorbed, and know the difference between HDL and LDLs, what they are, what they do, and from a health disease perspective, why are they important? Okay, that's it for transportation and absorption of digestion of fats. We'll see you in class.